Hello, my name is Arreh Jain. I'm a senior at Wazara High School and I conduct my research at University of Minnesota Twin Cities at Center of Magnetic Resonance Research, which is known for the world's strongest magnets. They use multiphoton imaging to collect their data. In this research, I used two-photon imaging to collect scans around the brain. Today, we're going to focus on the aspects of my research, especially about the blood vessels around the brain. I was inspired by my family history of neurological disorders as well as my interaction with others who have neurological disorders. I seek to improve the diagnosis, treatment, and cures of neurological disorders. Surprisingly, up to 1 billion people suffer from neurological disorders, and more than 50% of these people have to get hospitalized or go to the emergency room before they get accurate diagnosis. And functional movement disorders is a specialized neurological condition where people fall weak and they have to depend on their fellow people. Correct, detailed, and timely diagnostic imaging is critical for quantitative and qualitative analysis of disease progression. It is important to conduct this research on organisms in vivo as in postmortem the blood vessels start to shrink, making it hard to tease them apart. The Alexa Fluor 680 dye used in this research allows us to better see the blood vessels as well as differentiate the arteries and veins. The purpose of this research is to analyze brain scans, collect measurements to help assess the techniques of optimally mapping the fine-scale organization of arteries and veins in the brains of mice models using different lenses, dimensions, and slices in the scan. I hypothesized that the artery to vein ratio calculated from the living brain will be more accurate than the measurements taken from post-mortem samples. I will examine two photon optical imaging datasets which have submicron resolution and will enable for accurate segmentation and identification of arterioles and venules from the living brain. The goals of this research are to obtain measurements for length, the amount, the branching, and the spacing between arteries and veins. The materials used in this research include the Imaris software, which was used to observe the 3D models of layered scans of mice models obtained from two-photon imaging conducted by a grad student who pre-processed them to create clipping planes of blood vessels. Then, after the grad student obtained those scans of mice models using two-photon imaging, and then he coded them into 3D cube scans and color coded them into arteries and veins. I used the Imaris software to orient the cube scan and record the XY coordinates of the penetrating blood vessels which reach the surface of the clipping planes. Then using an Excel sheet, I calculated the distance between each and every blood vessel. Then I planned to use the MRS software to measure the distance, the size, and the diameter of the blood vessels. Here is the data that I have collected so far in my research. On the slide you can see there are arteries and veins that are turned on the cube scans. Here you can see the cube scan but with the combination of all the blood vessels with arteries and veins turned on in their scans respectively. And on the right you can see the layered scans obtained from the two-photon imaging. The 3D imaging for neurodegenerative disorders helps us view the arteries and veins and locate the problems in the blood flow. Although there could be possible sources of errors, I double check my work and verify the accuracy of the data. Some fun facts include that the cerebral cortex accounts for 80% of the brain's neurons. There is a misconception that dementia has a locus only in the hippocampus, which is not true as dementia is spread across the brain. Amyloid, Alzheimer's plaques, and aged vessels are everywhere in the brain. Dementia is usually associated with neuronal damage, and amyloid plaques kill the neurons also accumulate on the walls of arteries. This may limit the dilation and delivery of oxygen and nutrients to the brain, which power the brain. And it is difficult to tease apart the small arteries from the veins within the brain in post-mortem samples as the blood vessels start to shrink. In conclusion, tracing of blood vessels and from the scans taken in vivo using two-photon imaging is helping in their identification as they do not collapse and their true diameters can be measured. The goal is to develop normal baseline of healthy conditions so when a patient comes in, we can look at their scans and determine what is the problem they are facing and what is the cause of the same and eventually treat the problem. And the analysis of vascular anatomy, uh, the roles of vessels in fluid dynamics, and a summary of how their dysfunction can lead to neurodegeneration is also part of the goal. For future work, I will continue to analyze the available scans as well as do some scans on my own, and further research may guide the early detection of vessel pathologies in preclinical models of stroke, Alzheimer's, and aging. And other research about the circulation of blood in the brain may help with the detection of disorders and prevention of further damage using various medicinal and surgical approaches. Thank you.